My name is Pete Van Zant, and I'm an assistant professor at Birmingham Southern College. I've been at Birmingham Southern for about 8 years now, and I've been working on moths and moth projects for about 4 years. So I'm fairly new to the field, and that's kind of uncommon because a lot of people that study Lepidoptera have frequently started when they were pretty young. Okay, so how did I get interested on this particular project? Well, I guess I should go back to how did I get interested in moths in the first place. And what happened was my background is in studying herbivory, and I'm very interested in caterpillars and plant defenses. And I was working on a project in an area around here where these caterpillars were devastating these plants that everyone was really worried about. And so we were out trying to find the caterpillars, and the caterpillars would just appear, and we weren't finding any adults at all. So I said to the refuge manager, this was at a refuge, national refuge, and I asked the refuge manager if I could set up a sheet, because I'd heard this crazy idea about being able to set up a sheet and attract moths. And he said, sure, you can set up a sheet and have a bright light for them to collect them, but you have to tell me all the species that you find. And I said, that shouldn't be any problem because, there are these guidebooks and we can figure out what we've got. And I knew that there was going to be some issue of, or there were going to be some hard things to identify, but I had no idea what the diversity was like. At that site, we ended up collecting for, I think, 3 years, and we ended up finding over 300 species, different species of moths, and we estimated that there are over 400 in that area, and that's probably a very low estimate. It's likely that there are probably 1,500 species of moths at least, just in that one area. So as I was working on that project and trying to identify all this other stuff, I got really, really interested in the adults. They're not herbivores, but we don't know what they do. And the diversity is just overwhelming. And sometimes the abundance can be really overwhelming. There's just thousands and thousands of individuals. So I really got focused on doing studies on diversity of these different areas. And as I was working in the lab, putting things into the database and then shelving them, I started to notice what I thought was a trend in one particular relatively bland species, but it became a very interesting species to me because when we collected them at this refuge, which is a long ways away, the dates that I was noticing for the collection times were all about a week later than the dates that I would collect these moths on campus here in the city of Birmingham. And so I started wondering, well, is that phenomenon for real? And if it is, what could be causing it? And I just finished teaching an urban environmental studies introduction class. So I knew about the urban heat island from there, and I thought, well, maybe this is because of an urban heat island.
My name is Will Heminger. I am a rising junior at Elon University in North Carolina. I am from Des Moines, Iowa, and I have made my way down here to BSC to work in Dr. Van Zant's lab as a REU student, research experience for undergraduates. It is a program that is funded by the National Science Foundation based at Stanford University. My project is looking at moth phenology and how it is affected through heat island effects. We are doing that by sampling moth communities at 3 different sites. We've got an urban, an intermediate, and a rural location. That's kind of giving us like an urban-rural gradient that we can use to monitor what we capture at these different sites. So we're hoping to see results that show that most species of moths are emerging earlier here at the BSC than the urban location because the temperatures are warmer, and then where Turkey Creek is, that's our rural location, and then the temperatures are a little cooler there. That we'll see them come out earlier in the season. There are some species that will emerge earlier have their flight times beginning earlier at Turkey Creek, just because that's where, or we believe, that's where they're most suited.
Right here is our Ruffner Mountain Nature Preserve trap. As you can see, it's fully assembled and on. So as you can see, the battery is hooked up and powering the light. And right now we are actually attracting the moths. Inside the trap, though, is where we actually collect the moths. Inside the trap are 2 grates, which sort the moths based on size, for big and small. And also inside, sitting on the grates, are 2 containers full of ethyl acetate, which is our killing agent, that when the moths stumble into the light and eventually fall down into our trap by chance or due to fumes from ethyl acetate, the fumes then kill the moth, and then we can come in tomorrow and collect the whole trap and then take it back to our lab and sort the moths, and there we go. We have our data points.
What I've been doing recently has been counting focal species. We've got a certain amount of focal species that we're looking at, and then we're taking those, IDing them, counting them, and then we'll do some statistical analysis on it and make survival analysis curves that will show us where and when moths are emerging first. So the sorting usually, depending on what trap we're at, usually takes anywhere from 10 minutes to an hour. At Ruffner and Turkey Creek, where we get the most moths, it takes a while to go through and sort the focal species that we're looking at, just because there are far more individuals to go through. With the sorting, our focal species are ones that are easy to identify, or somewhat easy to identify, and then they're often pretty abundant. Our common ones, like Halosodota, Tesselaris, Nidata gibosas, what else? Doliacomas. Those are all probably our most abundant species, and what we'll use to run statistical analysis on and get survival curves. But for the sorting aspect, we separate the focal species from the rest of the species, and then count them and run our data from there.
The project that I'm working on now, the main project, is where we're looking at moths and their emergence times as we go from in the city, an urban environment, all the way out to a more rural environment. And there are several questions that you can look at. One is the diversity of moths in these different locations, and sure enough, we've been finding that the diversity is much lower in an urban environment, in an urban forest on our campus, than it is out in the country. But the main question that we've been focusing on recently is trying to see whether, first of all, is it warmer in Birmingham than it is in an outlying area, which is consistent with this idea of an urban heat island. And secondly, does that urban heat island, if it exists, affect the phenology or the flight times of moths? So that what we've been finding is that, sure enough, it's about, on average, 1 to 1.5 degrees centigrade warmer in the city than it is out at a place called Turkey Creek Nature Preserve, which is about 30 or so kilometers away. We've got an intermediate site at Ruffner Mountain Nature Preserve, another forested area, and at that intermediate site, the temperatures are about intermediate, and it's kind of on the edge of the city of Birmingham. And most of this difference is because of nighttime temperatures. So what happens with heat islands is that in the daytime the temperatures are about the same, because most of the temperature, the ambient temperature, is driven by incident solar radiation, so the sunlight is coming in. But what happens at night is these cities have been absorbing this sunlight and this intense radiation all day long. The concrete, the buildings, the pavement, and all these things are absorbing this radiation, and then at night they re-radiate it back out. So at night, Birmingham that's where the big difference happens. Birmingham is usually about 1 or so degrees warmer than it is out in a forested area away from the city. So what that's done to the moths is, we've been doing this project for about 3 years, and what that's done to the moths is it makes the many species fly a little bit earlier in the urban environment than they do out at the more rural environment. And our thinking is that this is because moths are ectotherms. Their metabolism, how fast they develop, when they emerge from the pupal stage, when eggs hatch, all these things, all these processes happen faster when they're warmer. And because the city is slightly warmer, moths do their thing slightly faster. The magnitude of this difference can be as little as 3, maybe 5 days, and at some times we've found that the magnitude of the difference is a couple weeks. So why this might matter, why this has been interesting to us, is because, first of all, this is a demonstration that urban environments are affecting not only the diversity of animals like moths, but it's also affecting their activity times or their phenology. The second thing that's of interest is not all species respond the same way. And the reason why that's interesting is because that about 1 to 1.5 degree temperature difference between the city and out in the country corresponds to one of the low estimates